Good morning. I am Dr. Yogesh, Professor and HOD, Department of Physiology, Sri Chamundeshwari Medical College Hospital and Research Institute, Channapatna. So the next experiment what we are going to see is the differential leukocyte count. I don't know, for some reason many of you are very much scared of this experiment, but if you ask me, this is the most easiest experiment to do. So what are the common mistakes here? First thing here is you have to know the name of the stain. That is the Leishman stain, what it contains and the function of each of the component. Eosin methylene blue, you already know what it does. The acetone-free methyl alcohol, this is where some of you are getting confused. So here what is happening is, once you have prepared a smear, when you add the Leishman stain 8 to 10 drops or sufficient enough to cover the slide, and you wait for 2 minutes. The first 2 minutes is called fixing time. What is happening in this fixing time? The acetone-free methyl alcohol, it is going to react with the plasma proteins present in your blood, and it is going to form a coagulum which is actually sticky in nature. So this coagulum is the one which is going to fix the smear, whatever you have made, to the glass slide. So only we are calling this as fixing time. And during this time there is no staining taking place. Then what is the next step? You add double the amount of distilled water or the buffer water, and you are going to wait for 10 to 12 minutes. Here what is happening? The stain is going to get ionized because you have added the buffer water, and the ionized stain only can enter into the cell and they start staining differentially. Acidic, like for the basic for acidic. So differentially it is going to stain it. Okay, so that is what is the difference, fixing time and staining time. It's always good to prepare 3 slides at least, and these 3 slides, when you're waiting for the staining time, what you do is, first slide wait for 10 to 12 minutes, second slide 12 to 14 minutes, third slide 14 to 15 or 16 minutes. Because sometimes the WBCs will take longer time to take up the stain. So if you, by chance, if you have not got the first slide, the last slide will definitely come, and there is no need to panic about it. Okay. The other things, when you look at the chart here, so it is ideal to get a nice good smear like this. Okay. Try to avoid the bands, pockets. Make sure whenever you are preparing the smear, the glass slide is clean. If the glass slide is not clean, it is going to leave pockets, and if you when you are making a smear, if you are giving a very small pause which you are not aware of it, you are going to get bands once again. So try to avoid all those, and it is good to know the ideal characters of a smear. Okay, that you have to know, and you should also know why you are doing this experiment. What additional information you are going to get regarding the patient. You have to know the conditions where the neutrophils are increased, neutrophils are decreased. Same thing for all the cells, eosinophil, basophil, monocyte, lymphocytes. At least know 1 or 2 examples. Physiological conditions, pathological conditions. Thank you.